A polypeptide is a linear molecule that consists of a chain of up to 20 different types of amino acids linked together by peptide bonds. The process of polypeptide synthesis for the hormonal protein, insulin, occurs in three stages: transcription, translation, and post-translational modification. The site of transcription is located in the nucleus of a pancreatic B cell, where the DNA is found, and it occurs because whilst DNA stores all the genetic information of the cell, it is too large to pass through the pores of the nuclear membrane, so a portable messenger, mRNA, provides a solution to this. Back to the DNA, it is composed of two elongated chains of nucleotides wounded in a double helix structure. Each nucleotide is made of a deoxyribose sugar, a phosphate group, and a nitrogenous base. There are four nitrogenous bases: adenine, thymine, guanine, and cytosine. The complementary base pairs are also held together by hydrogen bonds. The initiation stage of transcription of the insulin gene starts when the enzyme RNA polymerase II, with the help of proteins called transcription factors, attaches to a specific sequence at the start of the gene called the title box, located in the promoter region. This attachment, called a pre-initiation complex, unwinds the DNA, which causes hydrogen bonds between pairs to break and the separation of the two strands over a short length. The non-coding, or template strand, will be used as a template for the RNA polymerase II to synthesize the pre-mRNA, an unprocessed single-stranded molecule that carries instructions from DNA to ribosomes. The other strand is called the coding strand. During elongation, the transcription factors fall off as RNA polymerase II starts to move downstream, reading the template strand from 3' to 5' and synthesizing pre-mRNA from 5' to 3'. The enzyme finds floating RNA nucleotides and binds them with complementary template strand bases using temporary hydrogen bonds. Adenine and thymine bases pair together, while guanine and cytosine bases pair together. Also, when RNA polymerase II binds thymine to adenine, it will synthesize a uracil nucleotide on the pre-mRNA strand. The hydrogen bonds are temporary, as for every old pair that breaks, a new one is synthesized, creating a space around 6 to 10 base pairs long of unwoven DNA, called a transcription bubble. This is so that DNA behind the enzyme can rewind back into the original double helix shape. Covalent phosphodiester bonds between the RNA nucleotides in the pre-mRNA strand create a sugar phosphate backbone, helping the strand grow. The elongation process continues for the length of the gene until RNA polymerase II reads the termination sequence. The sequence is transcribed on pre-mRNA as a polyadenylation signal, telling the enzyme to stop transcribing, release the pre-mRNA molecule, and detach from the template strand. After, a 5' phosphate cap is added to the first RNA nucleotide, and a poly A tail is added to the last RNA nucleotide to protect the pre-mRNA from degrading. To form the mature RNA needed for translation, the pre-mRNA molecule undergoes RNA processing, where the non-coding DNA, called introns, are removed by an enzyme called the spliceosome. The coding DNA, or exons, are joined together to form mature RNA, and finally, the mRNA leaves the nucleus through a nuclear pore into the cytoplasm so that a ribosome can perform translation. During the initiation stage of translation, the small and big ribosomal subunits bind to the first codon of the insulin gene, forming a complete ribosome. A ribosome is composed of three sites: the A, P, and E site. The A site is the entry site of a new tRNA carrying an amino acid. The P site holds the growing polypeptide chain, and the E site is the exit site after the tRNA delivers an amino acid. During elongation, a tRNA, an RNA molecule involved in translating the mRNA, carries the amino acid GLY and enters the A site of the ribosome. Its anticodon CCU, a triple nucleotide sequence, binds to the complementary 3-nucleotide mRNA codon GGA. Any tRNAs with the incorrect anticodon are rejected and replaced with the correct one, in this case is UAC. The two amino acids in sites A and P form a peptide bond to keep the growing polypeptide chain in place. As the ribosome moves one codon forward, the tRNA in the P site is now in the E site and releases its amino acid GLY onto the tRNA now in the P site. The tRNA in the E site is released shortly, and the A site can accept a new tRNA according to codon pairing rules. Other amino acids include LLE, VAL, GLU, GLN, and CYS. The process ends when a unique stop codon sequence is read at the A site and can be either UGA, UAG, or UAA. A protein called a release factor binds to the stop codon in the A site, catalyzing the cleavage between the polypeptide chain and the tRNA. Finally, the polypeptide chain is released from the ribosome, and the ribosome dissociates into subunits, ready for a new translation process. To note, the polypeptide chain is called preproinsulin, and it needs to undergo post-translational modification in order to become mature insulin. The preproinsulin chain goes towards the rough endoplasmic reticulum, where it is pushed into the ER lumen to undergo the process. Polypeptide synthesis for insulin produces a primary, secondary, tertiary, and quaternary structure, as it has both the A and B chains needed to make the protein. Its primary structure is just a specific sequence of amino acids in the polypeptide chain that codes for an insulin protein. The secondary structure of insulin involves the folding of regions into 3 alpha helices, with the remaining regions being loops and turns. The tertiary stage involves the secondary structures interacting with each other and its environment, resulting in disulfide bond formations on the amino acid cysteine between the A and B chains. Disulfide bonds are strong covalent bonds that stabilize the protein structure. After folding, the proinsulin is packaged into vesicles and sent to the Golgi apparatus to undergo further processing. The quaternary structure involves the C-peptide connected between the A and B chains being cleaved by enzymes in the secretory vesicle, separating the 2 polypeptide chains which remain linked by disulfide bonds. Afterwards, the mature insulin protein stays in a vesicle in the cytoplasm of the pancreatic B cell until a signal is received to release it into the bloodstream when glucose levels are high. Polypeptide synthesis is important as it is a fundamental biological process where cells use their genetic material from their DNA to manufacture proteins. The creation of proteins from polypeptide synthesis occurs in all living organisms. Examples of proteins produced include insulin, enzymes, antibodies, structural and contractile proteins. Proteins are the building blocks of cells working together to perform the essential metabolic processes such as repairing tissues, building muscle, and aiding in reactions that produce energy to keep living organisms alive. Without the synthesis of proteins, life will cease to exist as the structures needed for both individual cells and the body will degrade and become irreplaceable.